Hi everyone. In today's video, I wanted to go through RNA translation. Now this is a continuation of one of my previous videos in which I went through DNA transcription. So RNA translation is the process that occurs after DNA transcription and is essentially when the RNA chain that has been formed after DNA transcription is able to then form a protein. Now, there are two main differences between eukaryotic and prokaryotic cells that define their translation processes. Firstly, in prokaryotic cells, as these cells contain no nucleus, the process of RNA translation can occur simultaneously with DNA transcription. However, as we all know, in eukaryotic cells, there is a nucleus present and therefore a nuclear membrane. Therefore, transcription must occur first and then translation must occur. Furthermore, in a video that I mentioned before, in prokaryotic cells, there are no introns present on the RNA strand. This means that the RNA strand does not have to go or undergo the process of splicing as a post-transcriptional modification. However, in eukaryotic cells, introns are removed, as these are essentially, and I say this in quotation marks, nonsense parts of the RNA strand. So, let's get to the process of RNA transcription in eukaryotic cells. Now, if we just take a step back for a second, we have just made the RNA chain, and we want to make it into a protein. So say, for example, the RNA chain is present as so, and this chain, now that it has been spliced, has undergone 5' capping and also has undergone polyadenylation, only contains the exons and these two features of a long poly-A tail, meaning having many, possibly even up to 125 adenine groups, and a methylated guanine on the 5' end. Now, this RNA chain is present within the nucleus, and we have to take it outside of the nucleus because that is where ribosomes are present, and these will be the main molecules which will be responsible for the synthesis of the protein. So how do we do this? Well, as you may know, the nuclear membrane actually contains small pores within it. Therefore, the RNA chain is able to essentially pass through these pores in order to reach the cytoplasm. So, in a very simple manner, this RNA chain is able to squeeze itself through the nuclear pores in order to reach the two small subunit, which I've denoted in this rectangle-like structure, and large subunit, which is this more triangular structure. Now, once the RNA chain has passed out of the particular nuclear membrane, it will reach the large and small subunits of a ribosome. The large subunit, just for reference, is actually made of RNA molecules, specifically rRNA, whilst the smaller subunit is made of protein, Now, it is important to remember that whilst the mRNA molecule is not bound to the particular ribosome, it is actually present as two separate subunits within the cytoplasm. It is only when the mature mRNA molecule binds to both subunits that it is able to combine and therefore allow for the process of translation to occur. So say, for example, we have our mRNA molecule now passing in to the particular ribosome. It does this in the cytoplasm, and therefore it can essentially allow for the process of RNA translation to occur straight away. However, after it has started translation, a very special process can first occur. There are special molecules that are floating within the cytoplasm called signal recognition particles, which I'll abbreviate to SRP. These particles are very important as they are able to allow for the direction of this particular forming protein to a particular part of the cell. Now let's say, for example, just as an arbitrary point, that the amino acid sequence is being formed from this rectangle. So we will have many amino acid molecules being formed in a bead-like structure. And just as a random, we might have these particular amino acids. What the signal recognition particle does is that it is able to bind to this forming amino acid sequence and therefore direct it to a particular part of the cell. Now let's say, for example, that the signal recognition particle recognizes that this protein should actually be formed on the endoplasmic reticulum. What it will do is then it will slow down the process of RNA translation. This will be until the ribosome actually is able to dock onto the endoplasmic reticulum. Then it will ensure that the ribosome comes in contact with a signal recognition particle receptor. Say, for example, we have the membrane of the endoplasmic reticulum here, and our ribosome is now docked onto it as so, with the particular mRNA chain now passing through it. The signal recognition particle, which I'll draw in a slightly enlarged manner, that is bound to the amino acid sequence, recognizes what we call a signal recognition particle receptor. This receptor will allow for the SRP to bind to it, and therefore these two molecules, which are the receptor and the particle, act as molecular matchmakers in order to allow for the amino acid sequence to find a particular protein translocation channel. So if we redraw our ribosome once again here with our mRNA sequence and amino acid sequence that is forming, once the SRP has been bound to the signal recognition particle receptor, a particular protein translocation channel is able to open up within the endoplasmic reticulum membrane. Now, the functions of this channel depend on what the particular function or manner of the protein is. If the protein is a water-soluble protein, then it will be able to pass straight through the protein translocation channel into the lumen of the endoplasmic reticulum. However, if the protein is a transmembrane protein, which I will denote in a different color, then it will not fully pass through the lumen into the lumen. However, it will pass in through a protein translocation channel, and it will eventually weave itself in and out of the membrane of the endoplasmic reticulum. However, I will explain this process further later. Now that this process has been established, I would like to go through further how the process of translation actually occurs. So, if we go back to our ribosome with its large and small subunit, we will actually see that there are three different sites that are present within the ribosome. So this bottom portion will denote the small subunit, and the large portion will denote the large subunit. Okay, now you may have noticed that there are three different sites present within this ribosome. These sites have special names. This is the A site, then there is the P site, and finally, then there is the E site. For example, if our particular mRNA molecule is passing from the side that denotes the A site and is going through in this direction, it will encounter some very important molecules. Firstly, as it does pass through here, what we call a tRNA molecule will come in contact with the mRNA sequence, where T stands for transfer. Now, as the particular mRNA molecule is able to then pass into the A site, which we actually call the aminoacyl site, the tRNA molecule, purely by random nature, is able to bind specifically with its anticodon sequence to the codon sequence present on the mRNA molecule. Now, what do I mean by that? Well, as I mentioned in the previous video with different mutations that are present, an mRNA molecule is read by a ribosome in terms of its codons. So say, for example, this is the first part of a particular mRNA molecule. Now, the start codon is always coding for methionine, which is AUG. Now, this codon sequence will be complementary to the anticodon sequence that is present on the tRNA molecule. Just as a reference, tRNA molecules are somewhat cloverleaf shaped, as so, and they contain the three particular codon sequences, which will be anticodon sequences, which will code for the particular amino acid. In this case, the tRNA molecule, if I was just to draw a small part of it, will contain the complementary to A, which is U, the complementary to U, which is A, and the complementary to G, which is C. As I mentioned earlier, this is a totally random process, and therefore, many tRNA molecules may first bind to this aminoacyl site before actually coding for the particular amino acid. So, as the tRNA molecule then ferries a particular amino acid, in this case methionine, to the ribosome, the start of the RNA chain can finally begin. From the aminoacyl site, the mRNA molecule moves to the P site, also referred to as the peptidyl site. This is when, if there is a continuation of the mRNA sequence, there will be another codon that is coded for by the particular RNA molecule. So, originally, as AUG was present in the A site, it will now move to the P site, as the mRNA molecule has shifted in this direction. Now, instead of AUG present on this side, there will be a new codon present of which the tRNA molecule will be able to code. Keep in mind that the original tRNA molecule that coded for methionine will still be present. However, now it will be present in the P site. Now, instead of methionine, there may be another amino acid that is coded for by this particular codon. Say, for example, we had UCA. This codon codes for alanine. Now, the complementary anticodon sequence for this would be AGU. As the particular amino acid is now alanine, the tRNA molecule will ferry this amino acid. Sorry, just to correct, this amino acid is actually serine. Now that two amino acids have been formed, the large subunit is actually also responsible for the formation of peptide bonds between amino acids. So a peptide bond will form between methionine and serine. Finally, the mRNA sequence will now move to the E site. This E site is where the tRNA molecule is finally able to detach from the mRNA sequence. It is also referred to as the exit site. Essentially, this AUG sequence, which has moved another place to the E site, will have its tRNA molecule removed. However, but the particular amino acid will stay on the chain. The particular sequence with serine will now move to the P site in the same way that methionine did. With the formation of a peptide bond. And finally, there may be another amino acid sequence that is formed in the A site with another random tRNA molecule, remembering that there is a unique tRNA molecule for each amino acid coding. There will be a random tRNA molecule that is able to code for a particular amino acid. In this case, if we take CAU, the complementary being GUA, there will be a histine. As this process continues on, the amino acid will start to grow. It is important to actually remember that the process of translation actually does not occur in a continuous manner. However, if we say the mRNA molecule is present as so, the large and small subunits move separately. If the large subunit is denoted as being on the top, it will move in a 5' to 3' direction, as so, and it will look something like this. Then, the small subunit will catch up so that they are equal once again. Then the large subunit will move to be as so, and the process will continue. So, this is the process of DNA translation. However, there is still an important part that is missing in our particular puzzle, and that is how an amino acid sequence is actually able to then bind to the membrane of the endoplasmic reticulum. Say if I redraw the membrane of the endoplasmic reticulum once again, as so, if, as I mentioned earlier, the particular protein is a water-soluble protein, then based on the particular protein channel, protein translocation channel that is present, the amino acid sequence will simply be able to pass through this protein translocation channel. However, transmembrane amino acid sequences are quite special. This is because they not only contain a water-soluble portion, but also contain a water-fearing or hydrophobic section. So if we say, for example, that this particular amino acid sequence has 2 water-soluble sequences and 1 water-fearing sequence, then it will essentially be able to weave itself in and out of the lumen of the rough endoplasmic reticulum. So let's see how this occurs. Essentially what happens is that each amino acid has a start sequence, and where there is a hydrophobic part present, there will be a stop sequence. This is because the amino acid sequence that has a water-fearing part does not want to enter the lumen of the rough endoplasmic reticulum. Once again, there will be now what's called an internal start sequence. And this process will continue on for however many stop and start sequences are required. So with this particular amino acid, if it is pushed into the endoplasmic reticulum membrane, then this particular part, which is hydrophilic, will move into the membrane. The hydrophobic part, on the other hand, will stay in the membrane of the rough endoplasmic reticulum and therefore dock itself here, and then the other hydrophilic part will be able to move into the lumen of the endoplasmic reticulum. This will be in somewhat of a weaving nature similar to a sewing machine. And eventually, if the amino acid sequence is long enough, it will form a structure like this. So this structure will continue on with the particular stop sequences and start sequences which are present. So this is essentially the process of RNA translation. We can have the amino acid sequences entering particular organelles in different manners. It occurs with the ribosomal units moving independent to one another. It involves three main sites, the A site, P site, and E site, in the movement of the amino acid throughout the ribosome. The amino acid can be moved to a particular part of the cell using signal recognition particles and signal recognition particle receptors. And the first step, however, in starting this entire process is the movement of this mRNA molecule from within the nucleus into the cytoplasm. And as we all know, the process between eukaryotic and prokaryotic cells is different. I hope you enjoyed this video. Thanks for watching.